The plasma membrane is the wrapper around the cell, so it would be like represented by this and like just everything around the whole cell. And then it is made up of phospholipid bilayer, which looks more like this. These are the phospholipids, and within those we also have glycolipids and cholesterol. So the glycolipids are sugar fats. They are also cell surface markers, and they keep the cells all stuck to each other. Those are represented here. These are the glycolipids. And then the cholesterol helps solidify the cell membrane, and it's in between the phospholipids, so those are represented by this right here. In the plasma membrane, we also have proteins. There are integral proteins and peripheral proteins, and that just describes where the proteins are. So peripheral proteins are on the outside, that is represented by this one right there, and integral proteins are built into the membrane, which would be represented here. Some functions of the proteins is transportation. There are channel proteins and carrier proteins. So a channel protein has a hole to let things pass through. This would be an example. It would pass through here, and the carrier proteins change the shape of things to let them through. There are also receptors, which is a way to get messages inside the cell. There's an intercellular adhesion, which keeps things together, and a cell-to-cell recognition, which helps the immune system recognize good and bad cells. The plasma membrane also has microvilli, which is this here. They are finger-like projections that increase the surface area of the cell. On the plasma membrane, there are also membrane junctions. There are tight junctions, anchoring junctions, and gap junctions. So tight junctions form waterproof connections between cells. Anchoring junctions are really strong connections in cells, and this includes desmosomes. And gap junctions form passageways between cells and allow material to move from one cell to another. Here we have the inside of a nucleus. There's a nuclear envelope, which is around the whole nucleus. We have in the middle the nucleolus, which makes ribosomes and contains chromatin. Here we have some more chromatin around the nucleus. This condenses to make chromosomes during cellular division, and the nuclear plasm, which is the whole inside of the nucleus.
Selective permeability helps regulate what comes in and out of the cell. Moving material across a membrane can be passive or active. Active requires energy, while passive does not. Simple diffusion and facilitated diffusion are passive ways of transportation. With simple and facilitated diffusion, molecules move from high to low down the concentration gradient. With facilitated diffusion, there are specialized membrane channels, and with simple diffusion, substances pass through phospholipids. Here are the diffusing particles, and they're going through in simple diffusion, and this is the carrier protein in the membrane, which is what the diffusing particles pass through in facilitated diffusion. Osmosis is movement of water from a higher water concentration to a lower water concentration through a semi-permeable membrane, which is what these lines are. Hydrostatic pressure comes from a force, which could be a push on the fluid, and then osmotic pressure comes from solute in a solution. Filtration uses hydrostatic pressure to push fluid through a filtration membrane. Tonicity is the measure of how much stuff is dissolved in the water. By determining this, you can determine which way osmosis will go. The more of something there is, the more water will dissolve into the cells because there's more. Here we have a hypertonic solution, and it is saltier on the outside than it is on the inside, which will cause it to go into crenation, or in other words, it'll cause it to shrink. and this is 10% sodium chloride. Here we have a hypotonic solution, and in this case the cell will swell up, and this is 0.01 sodium chloride. And here we have the isotonic solution, which is 0.9 sodium chloride, and nothing is happening because the water is moving in and out equally. Like I mentioned before, active transport requires energy, and in this case it burns ATP. Vesticular transport is movement of particles or even fluid droplets through the plasma membrane using the process of exocytosis or endocytosis. Endocytosis is moving stuff into a cell, and exocytosis is moving stuff out of the cell, such as waste or cellular products. Phagocytosis is a form of endocytosis, and this means cell eating. Bulk phase endocytosis is the internalization of a large amount of membrane into the cell and formation of a vacuole. Receptor-mediated endocytosis is when cells absorb things, such as hormones, proteins, and sometimes even viruses. RNA stands for ribonucleic acid and is singularly stranded. The role of RNA is to convert the information stored in DNA into proteins. mRNA stands for messenger RNA, and it helps with DNA codes for one protein. tRNA stands for transfer RNA, and that brings the proper amino acids to make proteins. rRNA stands for ribosomal RNA, and rRNA makes proteins. Transcription is making an RNA copy of DNA. Translation uses 3 types of RNA to make proteins.
So cytoplasm is a huge part of the cell. It's basically everything from the outside to the inside. So anything from the cellular membrane, which is the edge of the cell, to the nuclear envelope, which is the outside of the nucleus. Everything in between there is the cytoplasm. In the cytoplasm, you can find cytosol, which is the juice of the cell, and it holds all of these organelles in place, and it also provides a liquid environment for chemical reactions to take place inside the cell. The first organelle we're going to look at is the mitochondria, which are basically folded membranes, and they make ATP, which is energy that the cell uses. Mitochondria have their own DNA and ribosomes, which is similar to bacteria, and they divide by themselves inside the cell. Next we have the Golgi Apparatus, which packages, modifies, and changes, and then transports cellular products. It also works together with the rough ER and smooth ER. First, let's look at ribosomes, which are all of these little dots, and they also cover the rough ER, and they make proteins. They assemble proteins. It also works together with the rough and smooth endoplasmic reticulum. So first, let's look at ribosomes, which are all of these dots scattered around the cell. Those are free ribosomes, which just float around in the cytosol. There's also membrane-bound ribosomes, which are these ones that are stuck on the rough ER. Ribosomes make and assemble proteins and are responsible for the order of amino acids in proteins, and they use RNA to assemble the proteins amino acid by amino acid. So the rough ER, since it's covered in ribosomes, makes it a protein synthesis site. And then the smooth ER has no ribosomes on it, which is why it's the smooth ER, and it is a lipid synthesis site. So next we have lysosomes, which are these two things here, and they are like little stomachs for the cell. They're basically filled with hydrolytic enzymes and acid hydrolases, and they just kind of digest stuff for the cell. These yellow ones here are the centrioles, and around them, you can kind of see it drawn, is the centrosome, which is the area around the centrioles, and the centrioles actually make microtubules, which are these little blue things. Microtubules are the largest filament that is in the cytoskeleton, and they help make it up, and they form the mitotic spindle, which helps pull apart chromosomes during mitosis. Another filament that helps make up the cytoskeleton are intermediate filaments, which are basically, they're like the middle size, they're medium, and they attach to desmosomes and help resist pulling forces. And then the smallest type of filament that makes up the cytoskeleton, you can kind of see it all drawn around here, is microfilaments, which help the cell move around and change its shape. Peroxisomes are these two little ones here, and they are vesicles filled with oxidases, which detoxify harmful chemicals like alcohol. So they're kind of like the filters, kind of like what your liver does for your body, they do for the cell. Lastly, we have cilia, which are on the ends. They're kind of like fingers of the cell, and they help move stuff. So for example, in your respiratory system, they'll help move mucus out of your lungs and down your esophagus to your stomach. They also give the cell more surface area to absorb material. Another thing that helps cells move that's similar to cilia is flagella, and it's not pictured in this cell. The only kind of cell it's on is sperm, and it basically just helps the cell move. It kind of helps sperm swim around. Inclusions are little storage areas, and there's 3 that we talked about. The first is glycosomes, which are inclusions of glycogen, and they store carbs such as glucose. The second is adipocytes, which are fat cells where lipids are stored. And the third is melanin, which is the dark pigment in your skin, which protects your skin from UV rays. Interphase is not a part of mitosis, and it is the resting state of a cell before division. It is mostly an interphase, as you can see here, and mitosis is a small part of the cell's life cycle. This is the mitotic spindle, and it has not formed yet. And then in the nucleus, the DNA is still in a chromatin form. There's 3 stages. The first one is G1, which stands for growth 1, and the cell's just growing and being a normal cell. And if a cell is amitotic, that means that it's just in G1 its entire life, and it never divides. And an example of this is neurons. And then the second stage is S, which stands for synthesis, and in this stage, the cell copies its DNA and replicates it. And then in G2, it's growth 2, and in this stage, the cell grows and makes extra components to help it divide, which is like extra organelles and proteins, etc. And yeah, that's it.
Prophase is the first stage of mitosis. In this stage, the nuclear envelope breaks down, the chromatin condenses into chromosomes, which are these, and the mitotic spindle is formed.
Metaphase is the second stage of mitosis. In this phase, the cell ensures that it is ready to divide. The 2 sister chromatids of each chromosome are captured by microtubules from opposite spindle poles, and the chromosomes line up along the center of the cell.
Anaphase is the third phase of mitosis, and here the chromosomes split and get ready for division.
Telophase is the last stage in mitosis. This process separates the duplicated genetic material and the nuclear envelope reforms. The chromosomes uncondense and turn to chromatins, and the mitotic spindle goes away. Here you can see the cleavage furrow starts to form.
Cytokinesis is the last stage in the cell's life cycle, and it's when the cell physically splits. And as you can see, the mitotic spindle is not formed because it already split, and both of the cells have divided. And at the end of telekinesis, the cleavage furrow forms, but by cytokinesis, they actually split and the cytoplasm divides, and one cell turns to 2.